All right, thanks, Wade. Excellent talk. Do we have questions from the audience? I can maybe take, oh yeah, here we go. Sarah, please go ahead.
Yeah, thanks. Super nice talk. I was just curious about the role of the overdamped limit in this case. So that
Yeah.
was a bit too quick for me.
Yes. I mean, it's
Yeah, so okay, so my question is essentially whether you could also identify well, these general relations for the Surrey coefficient or something like that without drawing the overdamped limit.
um
So all the stuff that you show on the next slide, I mean, I guess all of these relations, because you would expect that something like the fluctuation dissipation relation could also hold in the underdamped case.
yeah, so the let's see. So we know that the fluctuation dissipation relation holds in the underdamped case. We've only, calculated it up to a given order in powers of the temperature because we're doing this low noise
Yes.
approximation, right? But so to the order that we've calculated, yes, we find that this local fluctuation dissipation relation still holds. The main reason we took the overdamped limit is because, again, we wanted to, match up with this sort of phenomenological description where we just have,
No, I understand that. I was just curious if that wouldn't be an opportunity now to also have an underdamped theory and still think about the Surrey coefficient and stuff.
Yeah, so I mean, it's, you can still have this potential of mean. So ultimately, this Surrey coefficient, there's two contributions. This alpha comes from this potential of mean force. This one comes from the fact that the diffusion coefficient depends on temperature and therefore on position. So you get this tendency for diffusion away faster from hotter regions and therefore a drift towards colder regions. And so those kind of get combined together in the overdamped limit.
Yes, right. Okay, thank you.
We have one more quick question. Okay. Well, thank you very much, Wade. And we'll go to our next speaker, Jeremy Guntoro.
Great, thanks.
I think, Wade, if you can stop sharing.
Yes, there we go.
Thanks.
Okay, I'm just gonna share my screen now. Sorry, give me a sec. Yeah, can everybody see my screen?
Yes.
Yeah,
I can see
perfect. Okay. So, hi everyone. My name is Jeremy. I'm here to present my project on information transduction in non-equilibrium templated polymerization, looking at specifically the interplay between heterogeneity and separation. So this is work done with my supervisor, Tom, as well as another student of Tom's group, Ben. Templated copolymerization, or polymer copying, is a fundamental process in biology. It encompasses the replication of DNA, as well as the production of protein and RNA, all very important molecules in biology. Accurate polymer copying is non-equilibrium. As an example, consider RNA copying. At any point in time, only a small number of monomers will be attached to the template. At some point in the copied process, a stable GC bond that is formed must eventually be broken. So you can't drive the process along simply by making the copy template bond very stable, because these bonds are transient, and breaking these stable bonds requires a free energy input. If, on the other hand, the bonds are not very stable, you can't bias towards accurate pairs. To illustrate the trade-offs involved in this non-equilibrium process, Poulton considered this simple model of polymer copying. Oh, sorry, let me get rid of that. It involves a fixed template with two types of template monomers and two types of copy monomers. It is a Markov model where a copy sequence is a specific state. So there is an exponential number of states with the template length. And the addition of monomers and the removal of monomers are both possible. These addition and removal steps are treated both as elementary reaction steps and obey a local detailed balance. At every step, copy template bonds are broken and reformed so that at any given step, only the tip copy monomer is attached to the template. So the effects of mismatches are temporary. This is similar to what you'd see in RNA copying. In Poulton's original model, homogeneity was assumed. So correct pairings 1,1 and 2,2 have the same binding energy, and the same with incorrect pairs. If you imagine at the very end, there are absorbing states to this Markov process where the copies detach. However, in Poulton's original work, most of what was considered was the infinite template limit. Thankfully for this model, the bulk product distributions are quite well behaved in this limit. I don't have time to cover this in detail, but we can actually relax the assumption of elementary reaction steps by coarse-graining the fundamental copying steps using methods in these publications. The coarse-grained system that results from this methods can then be solved exactly using the method in this publication. So back to Jenny's model, the homogeneous model has two free energy parameters, delta G pole and delta GTT. Throughout, we will measure our delta G parameters in units of kBt. Further, they will represent negative free energy so that more positive values imply more stability. So the first of these is delta G pole. Delta G pole is the only persistent free energy change that is incurred whenever a polymer is added to the growing product. And it is only removed when the associated monomer is removed. So as can be seen from this diagram, it shows up as a free energy change regardless of whether a correct monomer pair or an incorrect monomer pair is being added. It includes contributions, for example, from the formation of a nucleotide backbone. On the other hand, any copy template binding interaction will only last until the next monomer is added or until it is removed, at which point the bond would be destroyed. So this delta GTT term would only be incurred when moving, for instance, from a correct pair to an incorrect pair, and the opposite, vice versa. Whereas following up a correct pairing with another correct pairing will not result in any delta GTT term. The only term that would show up in the free energy changes there is this delta G pole term. Some key results from this model. There is an equilibrium value of delta G pole, which is negative log 2. At this equilibrium value, there is no accuracy in the product. So the entropy rate of the polymer sequence is ln2, which means there is a 50% chance of obtaining a correct or incorrect monomer pair at every given position. You can think of equilibrium as the point where the backward drive delta G pole exactly matches the forward drive obtained by the product entropy rate. So there is no copying velocity at this equilibrium point. So Poulton's original model was homogeneous, but biological polymer copying is actually heterogeneous. For instance, with RNA copying, the correct GC pairing might have a different binding energy from the UA pairing. Similarly, for incorrect pairs, UC might have a different binding energy from GA. So to extend Poulton's model to heterogeneous copying, we would now need a separate free energy parameter for every possible pairing of template and copy monomers. So given this heterogeneous model of polymer copying, I want to ask what the effect of applying heterogeneity to this separating model is. How would the results be different from homogeneous copying? So our first result compares the velocities of copying where the polymers separate as they're being copied. So this is the right, and copying without simultaneous separation, which is the left. That is, in this case, separation would only occur at the end of copying. So here we fix the binding energies of incorrect pairings at 0 and fix the binding energy of one of the correct pairings at 2, while the binding energy of the other correct pairing is varied as per this legend. We plot here the normalized velocity, which is just velocity normalized by some maximum value, against the non-equilibrium delta G pole drive that we put into the system. One result that is known in literature, the reference is shown here, is that for heterogeneous copying without separation, the velocity of copying reaches a near zero value, quite far away from the equilibrium point, actually. But in our case, where the copy simultaneously separates as it's being copied, we don't see such a tail of zero velocity. To understand why this occurs, we can think about the fluctuations in the free energy landscapes experienced at the equilibrium point. So for both these figures, we considered a fixed template and observed the difference in average free energy of the system at any given template index L compared to the average free energy for the template as a whole at equilibrium. Without separation, there is an accumulation of copy template bonds resulting in a random walk in the free energy landscape, which the net result is a is high barriers in the free energy landscape that would be difficult for the system to cross. But for the model with separation, fluctuations in this free energy landscape are much more bounded because of the transience of the copy template bonds. So this transience has a qualitative effect on the velocities that we expect to see as we approach equilibrium. The next thing we consider is the effects of heterogeneity on the error rates. We consider here Bernoulli templates, with the x-axis on these graphs being the Bernoulli parameter. We consider also two types of heterogeneity. On the left, we consider a case where the incorrect monomer pairs have equivalent binding energies, and the binding energy of one of the correct pairings is held constant while the other is varied as per this legend. On the right, the correct monomer pairings are made equivalent and the incorrect ones are made heterogeneous instead. If you look at the y-axis on these graphs, this is actually a measure of the heterogeneity on error. So the way it works is the numerator here is the average error, and the denominator is the control average error. This is the average error that we would expect if the monomer errors were independent of each other. The blue lines are the homogeneous cases, so these blue lines. And in this case, we indeed get that the measure is 0 as we expect. And what we find is that if you make the correct monomer pairings heterogeneous, we tend to get worse errors. And if we tend, because of heterogeneity, whereas if we make the incorrect pairings heterogeneous, we tend to get better errors as a result of heterogeneity. So this is not a result that was intuitive at first. However, we think we can explain this by considering the free energy landscapes experienced during copying. We can imagine that copying involves two different landscapes, either a correct landscape or an incorrect landscape, and any copying system would have a choice of moving between these two landscapes, resulting in a trajectory that could traverse the correct landscape for part of it and the incorrect landscape for the other part of it. Now, when we make the correct pairings heterogeneous, the correct landscape becomes harder to traverse and the system will bias towards the incorrect landscape. This results in the higher errors that we see. Whereas if you make the incorrect landscapes if you make the incorrect pairings heterogeneous, the incorrect landscape becomes rougher and harder to traverse. So the system will bias towards the correct landscape instead, decreasing the error rates. So based on these effects on error, we then considered making if we can make copying more efficient using heterogeneity. So Poulton in her original paper showed that there is an efficiency measure on the template condition entropy rate. So this is this HY given X. It is also straightforward from here to show that there is an efficiency measure on the mutual information. Note here that our efficiency measure is maximized over input distributions. We then plot these efficiencies maximized over the Bernoulli templates. We make the incorrect monomers more heterogeneous because this is the regime where we expected beneficial effects on error. The blue line in both figures are the homogeneous case, and the other lines reflect increasing levels of heterogeneity. We indeed observe increasing efficiency, both in the entropy rate efficiency and the information efficiency, with increasing heterogeneity in this regime, particularly when the driving is quite low in the negatives. So that concludes my presentation. I'd like to thank Tom, my supervisor, as well as Ben, my co-author in this publication, which contains everything from this work that I presented. I'd also like to thank other group members for helpful discussions. Thank you.